The URL for Soybean Knowledge Database, SoyKB, is SoyKB.org. The homepage has details about the latest developments, featured tools, news, and funding agencies. The toolbar at the top gives easy access to most features of SoyKB. The homepage also has a quick search tab for genes, mRNA, and metabolite search. The Contributors option under the About menu on the top shows information on the SoyKB development team members and collaborating labs. The latest development menu lists all the features and data sets added to SoyKB. It also keeps users posted on new features that are coming soon. The funding menu lists the funding agencies and all projects involved in the SoyKB funding. Under the Help menu, users can find details about our sign-up procedures as well as featured availabilities to the various levels of access. It also lists the details on how to request a higher access level. Users with VIP access levels can enter commands at the bottom of the Gene Card page. The Information menu shows the details of SoyKB architecture and information about the different modules in SoyKB. The Soybean Nutritional Facts link under the Information menu provides useful details and links to other sites providing information on nutritional benefits of soybeans. Users who have already registered can log in at the top right of the home page. Once logged in, the users have access to public and private datasets based on the user's access levels. The Search menu on the top can be used to access the four entities of SoyKB: genes, mRNA, metabolites, and SNPs. We will begin looking at the gene entity and searching for partial gene name glimma 17 gene 37. This search pulls out all genes on chromosome 17 that begins with glimma 17G 37. Genes can also be searched using gene families such as NAC. This search pulls out the list of all genes on all 20 chromosomes that belong to the NAC gene family. Each chromosome is listed separately. Genes can also be searched using annotated keywords such as reductase or using the COG domain ID such as COG 1502. This ID search pulls out the list of all genes on all 20 chromosomes that have the COG 1502 domain in it. Each chromosome is listed separately. Clicking on one of the gene names brings the users to the respective gene card page. Genes can also be searched by entering a chromosome number and specifying a nucleotide range. Any genes falling in these coordinates are returned as separate lists for mRNA and CDS entries. They are individually linked to their gene card page. We will begin by looking at the gene card entry for glimma 06G 45700.1 gene. The top of the gene card page shows the gene name and gene family information, followed by the gene model information with the UTRs, exon and intron positions shown as different colored blocks. This is followed by starting and ending nucleotide positions, as well as strand information for the gene, including individual exon and UTR positions. Clicking on the C button under the Genome Browser column takes the user to the locally set up Soybean Genome Browser at MU that focuses on the respective gene or axon and UT regions. In the Genome Browser, users can visualize the experimental data of the selected region of the chromosome. The Sequence Information tab on the gene card page shows the cDNA, CDS, and protein sequences for the gene. The domain information from COG, Panther, and PFAM are also available for genes with known functional annotations. The Pathway Viewer is enabled in the gene card page and can be viewed via the Open Pathway Viewer button. This opens the Pathway Viewer in a new tab. The gene is highlighted by a red blinking circle on the pathway for easy identification. The protein 3D structure can be viewed via the Embedded Structure Viewer. The 3D rotating protein structure can be moved and rotated by clicking the mouse and dragging it from side to side. The sequence-based experimental data from EST 5' rate and full-length cDNA datasets are also available for genes. The 5' rate data is linked to display details about the accession ID, experimental condition, sequence, and sequence length. The transcriptomics RNA-seq experimental data can be visualized under the Experimental Data tab. The RPKM expression abundance values can be seen by moving the cursor over the graph. Microarray data from leaf and root are a private dataset. It is available for users to view based on access levels. The corresponding Affymatrix probe name is displayed at the top of the graph. The microarray dataset for tissue and stress are publicly available and have 25 and 99 conditions respectively. Users can click on the View Subgraph button next to each condition to look at the data for the selected condition. This allows users to take an in-depth look at data from one condition. Proteomics data also shows the individual peptides at the top of the graph. Individual experimental conditions can be viewed using the subgraph feature. The proteomics dataset is also available for current seed conditions. At the bottom of the gene card page is a tab for references. This tab links to the literature articles about the genes as well as the protein names from PubMed. These articles are dynamically retrieved and represent the latest information from the respective genes. The articles are linked to their respective PubMed pages for easy access. Now we will look at the miRNA entity. miRNA can be accessed using the search menu. The search menu supports partial name searches. The miRNA card page lists the details about the miRNA sequence, miRNA family, expressions in small RNA libraries, and the predicted target genes in the soybean. The target genes are linked to their respective gene card pages. The user may also choose to browse the entire list of all available miRNAs. Each miRNA is clickable. Now we will look at the third entity, which is metabolites. Metabolites can be accessed using the search menu. Metabolites can be searched with full or partial names. If multiple matches are identified, the users can select the most suitable one. This brings up the metabolite card page. The metabolite card page shows information for name, mass to charge ratio, retention times, as well as a link to the pathway viewer. For ease of detection, the pathway viewer highlights the metabolites with a red blinking circle on the pathway. With the metabolite highlighted, the pathway can be seen in full screen as well as saved in an image file. Data from the HMDB database is also available on the metabolite card page. This page provides information including the chemical formula, the IUPAC name, the molecular weight, and SMILES formulas. Metabolite expression data from the LCMS or the GCMS experiments can be viewed as graphs and subgraphs on the same page. Next, we will look at the SNP entity, which is also accessible under the search menu. SNPs can be searched using a chromosome number and the nucleotide coordinate range or by entering the SNP ID. The SNP card page displays the SNP ID, chromosome position, consensus, and reference base information, read depth, and read equality supporting the SNP. Multiple genes can be queried at the same time using the search feature for groups of genes. A user can enter a list of genes and select the suitable ones from any multiple matches. Each gene name at the top is linked to its respective gene card page. Next, users can adjust the correlation cutoff used to display the information. A pie chart of the gene family distribution for multiple genes and the names and numbers of genes in each gene family can be seen by browsing over the pie chart. Clicking on the transcriptomic dataset links show the correlation coefficients based on select cutoffs, followed by the graphs of RPKM for multiple genes. The proteomics dataset is also displayed in a similar fashion. Each gene is colored in a different color for easy detection. For microarray datasets, the gene names with respective Affy-matrix probe names are displayed on the top of the graphs. Users can move the cursor over the graph to see the expression values for the individual data points. Sequence-based data from ETS5, prime rate, and full-length cDNA are also available with the links at the bottom of the multiple query genes. Each dataset shows all accessions IDs, sequence information, and condition details for the respective data. Multiple metabolites can be queried at the same time using the search features for groups of metabolites. Users can enter a list of metabolites and select the suitable ones from any multiple matches. Each metabolite name at the top is linked to its respective metabolite card page. The experimental data for all metabolites from LC-MS and GC-MS experiments are linked along with details about the respective experimental conditions and the labs which generated the data. The graph shows the data for roots in uninoculated and inoculated conditions. Clicking on the link for CLMS data shows the multiple metabolite expression plotted together for the different conditions. Metabolites can also be searched using the Search by Experiment feature. Users can select experiment, tissue, condition, and polarity type. The selected metabolites identified in this combination of experiments will be listed. Each metabolite is linked to its metabolite card page. Another feature of SoyKB is to be able to browse gene families. This feature is available under the Browse menu. Users can select from a list of gene families to choose from, including transcription factors, cytochrome P450, and others. They can further select a specific family amongst transcription factors from the pull-down list. All genes in that particular family will be listed according to the chromosome numbers. Users can navigate to the gene card page by clicking the gene name of interest. SoyKB has a feature for protein 3D structure visualization. This is available within the browser menu. Users can search for a gene name and then see the predicted 3D structure. The click and drag function of the mouse allows the user to rotate the structure in 3D space. The PDB formatted 3D structure files are also linked on the same page for the users interested in seeing the details of the 3D structure. The Tools menu on SoyKB provides access to many useful tools, including the Affy-matrix probe ID mapper. Users can enter the full or partial Affy-matrix probe name, and the tool identifies the corresponding genes. It also links the genes to the gene card pages, which users can see the microarray expression data. The next tool is the Gene Pathway Viewer, which allows users to enter a list of multiple genes and maps all of them at once to the multiple pathways. The different genes in the list are all highlighted with red blinking circles. The drop-down pathway list at the top of the page can be used to select and view other pathways by clicking on the View Pathway button. The Metabolite Pathway Viewer has similar functionalities and allows users to query a list of multiple metabolites at one time. The different metabolites in the list are all highlighted with red blinking circles. The pathways can be seen in full screen mode for better viewing. SoyKB also allows its users to download both private and public data for multiple genes based on the user's access level. Users can enter a list of genes and select a data type to download before clicking the submission button. This saves the results to a file that can be downloaded by the user. Users can also contribute their own data to SoyKB using the Upload option under the Data Files menu. Users can choose a formatted data file for submission of different types of data. The gene family information can be submitted using a 2-colon tabbed delimited format. Users can also enter their contact information, including name, address, and affiliation in the text fields, as well as select the data type format prior to submitting the information.